Ladies and gentlemen, thank you very much for giving me the opportunity to give you an update on our data that we have collected recently with idebenone. The trade name that we're using in Europe is Raxone, so I will use both names interchangeably in my presentation, on respiratory outcomes in patients with Duchenne muscular dystrophy. So what I'd like to do now is, first of all, explain very briefly the medical need for the treatment of pulmonary function loss in patients with Duchenne muscular dystrophy. Then I will briefly touch on the mitochondrial pathology involved in Duchenne muscular dystrophy and the rationale to use this molecule idebenone as a potential treatment. I will then give you a brief overview about our comprehensive development program that we have conducted in the past 10 years. And I will dive into a little bit more detail in the outcome of a phase 3 trial that we have conducted already a little while ago in patients not using concomitant glucocorticoid steroids. We will talk about efficacy data on expiratory and inspiratory function, and we will also talk about observations that demonstrate that the treatment actually reduces bronchopulmonary complications such as airway infections. And then I will introduce you to a new trial that is about to start, both in the United States and in Europe, for patients using concomitant glucocorticoid steroids. And the last, I will give you a brief update where we stand in our regulatory processes. So just to put the whole thing into context, the company is developing this molecule, idebenone, Raxone, as I said, in 3 indications, all associated with mitochondrial pathology. The drug is approved in Europe for the treatment of a disease that causes blindness and is also caused by mitochondrial pathology. And we are running a phase 2 program with the NIH in primary progressive multiple sclerosis, again a subform of MS, which is associated with mitochondrial pathology. Of interest today, obviously, is Duchenne muscular dystrophy, and I will illustrate the fact that also in Duchenne muscular dystrophy, a mitochondrial component contributes to the disease progression and our approach to actually counter that. Let me just introduce you to the medical need for effective treatment in respiratory or pulmonary illness in Duchenne muscular dystrophy. As illustrated here, with age, medical complications affecting the respiratory capabilities of the patients become more and more evident. These include ineffective cough, nocturnal hypoventilation, sleep disordered breathing, and ultimately daytime respiratory failure. With age, DMD patients develop cardiac and respiratory complications, which together frequently become life-threatening. So how do we measure in routine practice pulmonary function? Typically this is done with a spirometer, and what you do is you record what is called flow volume curves, illustrated here. In the green curves in the middle, on the B, and to the right on the C, you see 2 age-matched individuals. On the B you see a DMD patient, and on the C you see a healthy boy. And what you see is that the 2 curves are not the same. The DMD patient curve is actually smaller in both axes, which means that there's a reduction in peak expiratory flow, that's on the y-axis, that's the speed with which you can exhale air, and a reduction in lung volume, measured as forced vital capacity, or FV1, forced expiratory volume in 1 second, and that's on the x-axis. So both dimensions, flow and volume related parameters, become progressively reduced in patients with Duchenne muscular dystrophy. This is illustrated here in a qualitative way, and these are data from the Synergy network, which I gladly present here, and over 330 boys. And what you see is here 2 of these measurements overlaid on each other over the age, which is shown on the x-axis. And you see that both peak expiratory flow, the flow parameter, and the volume parameter both decline in a comparable and parallel way starting from around 10 years of age onwards. And by the age of around 20, 22, 24, it reaches levels which are very low, representing only 30% of what a normal individual would have at that age. So we have now 2 parameters, expiratory function parameters which decline in parallel, and which is a cause of medical concern in these patients. The fact that treatment of pulmonary function is relevant for patients has been demonstrated by PPMD's efforts to collect data from the community. And you may be aware that there was a patient-centered benefit risk survey conducted and completed by PPMD, which illustrated that treatment of pulmonary disease, like the preservation of effective cough or the prevention of airway infections, was highly prioritized by patients and caregivers alike. So what is the urgent medical need? Well, in fact, that's illustrated here. You see two curves. The blue dotted curve is actually the use of steroids in Western countries like the United States, Canada, and Western Europe. And you see that most of the boys are taking steroids due to the fact that steroids have a beneficial effect on multiple aspects of disease progression. However, with time, and with the emerging side effects in each of these patients, and the fact that patients go into the wheelchair at some point in time, the use of steroids is decreasing. And there's recent data published again by the Synergy group that indicates that about 40% of patients 10 years and older are no longer using glucocorticoid steroids due to the side effect profile. But this is exactly the time when the respiratory function loss kicks in. So we have a dilemma here. We should use steroids because they're beneficial. On the other hand, with time, the tolerability is becoming problematic. And exactly at the time when patients become non-ambulant and the loss of respiratory function becomes evident. This is where we would like to position the use of our drug in the treatment of DMD. Let me just park here for a second and explain you why we think it actually could work in DMD, and we have data for that. This is a highly illustrative and simplified slide, but it makes the point that downstream of dystrophin deficiency, when dystrophin is lacking in the patient with DMD, there are numerous biochemical cascades that kick in. One of them is actually caused by little ruptures in the membrane of muscle that, following cycles of contraction and relaxation, leads to an uncontrolled influx of calcium. And this calcium overload eventually causes malfunction of mitochondria. These are the powerhouses of the cells, providing the cell with energy. In addition, lack of energy also is coupled with an increased level of reactive oxygen species. These are cell-damaging, very potent radicals that can damage other molecules in the cell. So the message here is, downstream of dystrophin deficiency, we have cascades, biochemical cascades, that involve mitochondria and actually hinder the mitochondria to produce the energy that is normally needed for the muscle to function well. Now, our molecule idebenone actually has been demonstrated by a series of non-clinical and animal experiments. It actually can increase the production of ATP, which is the energy source of muscle cells and other cells, and reduce the production of reactive oxygen species. So essentially, it is self-protecting in a situation where the primary defect, the lack of dystrophin, is not cured. We do not touch the dystrophin molecule, but we act on the downstream consequences of dystrophin deficiency. Now, this is a slide that shows you the extensive clinical program that we have conducted so far and we continue to conduct. We have conducted a phase 2 program, which we call the Delphi program, that started back in 2005. And this was an exploratory trial that helped us to find endpoints and also refine our strategy with the development program going forward. I would like to focus the rest of my presentation on 2 phase 3 programs. One is already finished, that's the Delos trial, and one is about to start, that is the Ceteros trial, and I will talk about both. Now, as also illustrated in this slide, we have 2 areas, a gray shaded area and a orange shaded area. And this should illustrate where the trials are actually conducted with respect to patients' use of steroids. In the Delos trial, this was the first trial that's completed, we have enrolled only patients that were not using concomitant steroids. In In the new trial, the Steroids trial, we will actually allow patients who are on steroid treatment. So I will illustrate both of these aspects. So the Delos phase 3 trial is already complete for some time, and actually was published in 2015 in Lancet. And I will only be very briefly summarizing some of the findings. So the patients that were enrolled in that trial were aged 10 to 18 years. There was no restriction to any mutational status, but the patient had to be off chronic steroids for at least 1 year prior to enrollment. As a consequence, 92% of the patients were already in the wheelchair when they started the trial. We randomized and treated 64 patients, of which 31 were on active treatment for 1 year and 33 were on placebo. The average age of the patients participating were 14 years. Now this is a slide that summarizes a lot of data from the expiratory efficacy outcomes that we have investigated. Again, the peak expiratory flow or the lung volumes, FEV1 and forced vital capacity. You see the data for the 6-month and 12 months time point. And on each line you see a dot and a little line, left and right to the dot, and this is what is called the effect size and the 95% confidence interval. Whenever you see a dot to the right of this dashed line, which is going vertically, this indicates that the drug was in favor, benefited Raxone over the placebo. And you see that all of these endpoints, they're all on the right side of the dashed line, indicating that the drug worked in any of these endpoints, both at 6 months and 12 months time points. And for those of you who are more into the numbers, you also see the p-values for statistical significance. Now, as I said, these data have already been published and they have already been discussed in the community. And we would like to focus on a few more recent analyses that we have conducted. Starting with inspiratory function. Because remember, for respiration you need both. Inspiration and expiration. I've shown you now the data for expiration, now we talk about inspiration. That's a methodology that has not been used much until very recently in Duchenne muscular dystrophy. And it actually asks the patient to do what is called a tidal breathing, and then we measure the maximum inspiration of the patient. I will illustrate that to you, so just bear with me. I do now what the maneuver actually is all about. So you initially breathe normally. And then you suck in with maximum force you can. And this is the maximum speed with which a patient can inhale air. And what you see is the difference between what is called on the slide VI max T, which is the normal tidal breathing, and the maximum effort that is seen in the forced vital capacity. And the difference, the ratio, is important because it tells you how much reserve a patient has to increase the velocity to inhale air. And with patients, this safety margin is actually getting smaller and smaller and smaller over time. So we measured that in our trial. And you see here to the left, there's the maximum inspiratory volume, the velocity, the speed. And you see in orange, active treatment group, and in black, the placebo group. And to the right, you see this reserve, which is a safety margin that actually patients have. And you clearly see that there's a larger safety margin in patients having received active treatment compared to those on placebo. Again, for those of you interested in the numbers, you see the estimated treatment effect and the p-values on the bottom of the curves. In the inspiratory flow reserve was actually statistically significant between active and placebo at week 52. So here we have now both. We've seen an effect on expiratory function and inspiratory function. But you can still ask the question, well, what is the relevance for the patient? I give you 3 examples about the relevance and what actually for the patients. And one is, for instance, peak cough flow. We all know that cough is important, maintenance of cough is important for the patients. And there is a threshold of 160 liters per minute, which is commonly regarded as a minimum threshold of peak cough flow that is required to clear the airways effectively. If the patient falls below 160 liters per minute, the risk of airway infection is increased. Now, although the numbers are small, but we had 26 patients in the active treatment group and 33 patients in the placebo group who were above 160 liters per minute at the start of the trial. 6 patients in the placebo group fell below this clinically relevant threshold in the placebo group, and only 1 in the active treatment group. Again, numbers are small, but it shows that what we have measured with the instruments actually can also be seen in changes in peak cough volume. But further, we also looked at the number of airway infections and related bronchopulmonary complications that were reported in the trial as side effects. And we saw a clear signal in the placebo group for more of these side effects. So we see in the placebo group there were 17 patients reporting 28 bronchopulmonary disease events, so complications like airway infections, compared to only 6 subjects in the active treatment group reporting 7 events. To the right, you see the hazard ratio. This is the cumulative risk of a patient experiencing bronchopulmonary disease in the trial. And there was a 72% reduction, which was clearly statistically significant, in the patients receiving active treatment in experiencing any of these bronchopulmonary disease complications. The number of days patients in the trial were affected by these diseases were 220 days in the placebo group compared to 82 days in the active treatment group. And I'm glad to say that this data have recently been published in Neuromuscular Disorders for those who are interested in more detail. We then went on and investigated actually whether there's a change in the use of antibiotics normally used to treat these bronchopulmonary disease events. And again, there are fewer patients in the active treatment group needing antibiotic treatment compared to the placebo group. The cumulative days treated with antibiotics in the trial were 105 days in placebo and 65 days in the active treatment group. Again, clearly reducing the risk of needing antibiotic treatment due to complications of the airway and bronchopulmonary complications. So let me summarize Santhera's outcomes. So the phase 3 trial met its primary endpoint. I think I didn't mention that, but I should have done it. This is so far the only trial that met its pre-specified primary endpoint in Duchenne muscular dystrophy. We have supportive evidence for efficacy from secondary endpoints, and we demonstrated a consistent treatment effect on expiratory and inspiratory function. We also have supportive evidence for efficacy in clinical relevant responder analyses, such as those the number of patients maintaining effective cough. And we have seen demonstrate clinically relevant impact on bronchopulmonary disease and antibiotic use. Now, switching gears a little bit, these were all data I've shown you in patients who have not taken steroids. Now, you may ask, what is in patients on steroids? Today, we don't have sufficient data to make any statement about that. But we are about to start a new trial, which is called the Steroids trial, which is specifically for the enrollment of patients who take concomitant steroids. And I will talk a little bit about that trial in a minute. The reason is the following. Although, if you are taking steroids and patient clearly benefits from steroid use, there are now data that have been collected by the Synergy group here again that clearly demonstrate that patients are protected on steroids for about 2 to 3 years longer than patients not taking steroids, of falling below a clinically relevant threshold of pulmonary function decline, which is commonly determined as 80%. So as you see here in these little arrows, patients that are taking steroids are benefiting for about 2 to 3 years longer in maintaining effective respiratory function. But once this decline has started, you also see that the rate of decline and the slope of the 2 curves is actually parallel. So there is still a need to also treat patients on steroids who experience pulmonary function loss. And that would be the objective of that trial. So clearly the objective is to assess the efficacy of IDEP compared to placebo in slowing the loss of respiratory function in patients receiving glucocorticoid therapy. That is a trial that will enroll 260 patients roughly, and patients have to be on stable steroid dose. We look again at respiratory function endpoints with forced vital capacity at a primary endpoint. Interestingly, the trial does not have a requirement for age. There's no age category involved here, but the patient has to be at a starting decline phase of respiratory function and should have still some residual respiratory function left, which is commonly defined as forced vital capacity between 30% predicted and 80% predicted. It's a 1.5-year trial. It's a randomized placebo-controlled trial, and every participant is subsequently invited to in an open-label extension trial where all patients get active for at least 1 year until the approval of the drug. So this is the rough outline. If you would like to have more information about the trial, we have a poster that is here shown at the presentation, and my colleagues will be there to answer any further questions on the trial design. Now these are the investigators. I am happy to have Hank Meier. He's sitting in the front row. He's the lead investigator for that trial in North America, Children's Hospital Philadelphia, and my colleague Boise in Leber, Belgium, will be the lead investigator for Europe. The trial will be conducted in about 50 centers, equally split between the United States and Europe. Currently, we have a list of sites that have been selected, or cities that have been selected. We are still updating this, and you will see an updated list of potential trial sites on clinicaltrials.gov. I expect that actually the trial will be visible on clinicaltrials.gov by the end of this week. Now, I would like to end with a regulatory update. We have, in Europe, starting with Europe, we have recently submitted a marketing authorization application for our drug with the EMA, European Medicines Agency. The MAA, the application has been validated and the review process has started, and we expect a decision by the CHMP, which is the decision-making body of the EMA, in the first quarter of 2017. Remind you, the drug is already approved in Europe for another indication, so this is actually what is a line extension and an additional indication added. In the United States, we have submitted a proposal to the FDA for an accelerated approval based on the existing Delphi and Delos trials and the Citrus trial as a confirmatory trial, and we are waiting on feedback from the FDA in our discussion. The intended indication at this point from the current data set would be patients in whom respiratory function has started to decline and who are currently not taking glucocorticoids. The indication would include, however, patients who previously were treated with glucocorticoids and have stopped to take them, in whom glucocorticoid treatment is not desired, not tolerated, or contraindicated. So in summary, how we see the positioning of Identin Raxone for the treatment of DMD. There's a sizable proportion of patients who cannot tolerate glucocorticoids at the time when pulmonary function decline is actually becoming evident. And therefore there's an urgent medical need to slow down the decline of pulmonary function in these patients. The available data demonstrate that the investigational drug Raxone slows the loss of pulmonary function in patients not using steroids. And the initial application for approval will be in this patient group. Raxone was tested in patients without restriction to specific mutational or disease status. So specifically whether or not you're ambulant or not is not a question here. The drug is an oral medication taken 3 times a day, it's 2 tablets 3 times a day. And the new study that we are about to start will investigate the efficacy of the drug in pulmonary function outcomes in patients using concomitant glucocorticoids. And with this, I would like to thank you for your attention.